Insect populations don't just exist at a constant number of individuals. They change. Numbers rise, numbers fall, but why? What governs these fluctuations? Is it the environment, food availability, predation? Maybe something about the organisms themselves? When it comes to insects, these questions aren't just theoretical. Insect populations can and do shift dramatically, sometimes predictably and sometimes not. Today, we'll break it down. We'll look at both the biotic and abiotic forces that act to shape insect population dynamics. Let's start with the abiotic factors. These are things like temperature, precipitation, and soil conditions. These don't just influence insect survival, but they can sort of set an upper and lower boundary for population dynamics. Temperature is one of the most fundamental abiotic factors influencing insect populations. But the relationship isn't simply about being too hot or too cold. Many insects exhibit degree-day dependence, meaning their development is tied to the accumulation of heat over time, rather than simply the passage of days or the temperature on any given day. In essence, it's calculated as the number of degrees above a species' developmental threshold that accumulates day by day. One well-documented example of degree-day dependence comes from the Colorado potato beetle, which is a major pest of solanaceous crops like potatoes and tomatoes. The beetle requires about 60 to 90 degree days above 10 degrees Celsius to complete its development from egg to adult. A cold spring could thus slow the life cycle considerably. But in warmer years, the beetle can complete its life cycle more quickly, and might even fit in an additional generation, which would significantly increase population growth, and along with it, crop damage. As you might expect, this feature of multiple generations per year can significantly be impacted by climate warming. For example, the fall webworm has what we call a flexible volatinism trait, meaning the number of generations that's produced per year can shift based on temperature and on geographic location. In cooler regions, they're univoltine, meaning one generation per year. But in warmer climates, they can become bivoltine, or even multivoltine, which is 2 or more generations per year. What's interesting, though, is that this shift to warmer temperatures isn't universally beneficial for insects. Some cold-adapted species, especially those with chilling requirements for diapause termination, which is a period before they start the next phase of their life cycle, they can be at risk for phenological mismatches. Take the winter moth. Traditionally, its eggs hatch in the early spring, coinciding with the budding of host plants like the oak tree. However, warmer winter temperatures have led to earlier egg hatching, sometimes even occurring before the host plants have produced sufficient foliage, resulting in higher larval mortality starvation. Precipitation can play a similar role, but often with more indirect effects. Drought conditions, for example, can increase insect outbreaks like what we see with bark beetles. When coniferous trees experience water stress, they can produce less resin, which is a key defense mechanism against bark beetle attacks. The result? An uptick in bark beetle infestations coinciding with droughts that can lead to widespread forest damage. Even something like sunspot activity has been implicated in the rise and fall of insect population abundances. Sunspots are dark, cooler regions on the sun's surface caused by intense magnetic activity. But when they form, they're often accompanied by bright, hot areas called faculae that actually result in increased overall solar radiation. During periods of high sunspot activity called solar maxima, Earth receives slightly more energy, leading to slightly higher UV radiation levels. Some evidence suggests that this can make winters in some areas a little milder. Conversely, low levels of sunspot activity, called solar minima, means lower UV radiation levels for Earth, which can contribute to slightly cooler winters in some regions. For something like the autumnal moth, which overwinters in the egg stage, these colder winters during solar minima can have the potential to be a limiting factor for population growth, because a higher proportion of eggs are lost due to freezing in cold winter temperatures. However, increases in times of solar maxima result in a higher proportion of eggs surviving, and over successive favorable years, populations can see dramatic increases, even leading to outbreaks that often start during years when sunspot occurrence is low. So, temperature, precipitation, climate cycles, they all set the stage. But abiotic factors alone don't determine insect population dynamics. To understand the full picture, we need to look at biological or biotic factors. Here we have bottom-up effects from plants and top-down effects from natural enemies. At the most fundamental level, bottom-up forces include things like the quality and availability of plant matter. You would think that when plants are doing really well, that insect herbivores would also do really well. But that's not always the case. Here we have two different hypotheses to explain how plant condition influences herbivore populations. There's the plant stress hypothesis and the Plant-Vigor hypothesis. Rather than being mutually exclusive hypotheses, they sort of highlight different ecological strategies amongst insect herbivores. The plant stress hypothesis suggests that herbivorous insects often benefit from plants experiencing physiological stress, particularly drought. Under water stress, plants frequently reallocate resources, resulting in increased concentrations of soluble nitrogen and amino acids in their tissues. Since nitrogen is a critical limiting factor for insect growth and development, this enrichment can make stressed plants a better food source for certain herbivores. And stressed plants may also reduce their investment in chemical or structural defenses, making them more vulnerable to attack. One example of this would be psyllids feeding on eucalyptus in Australia. Outbreaks of these sap-feeding insects frequently coincides with periods of drought, when trees experience that physiological stress. The elevated nitrogen content in drought-stressed eucalyptus leaves leads to higher psyllid survival and faster population growth, sometimes resulting in large-scale defoliation events. On the other side, we have the Plant-Vigor hypothesis, which is a bit of an opposing view. It argues that many insects actually prefer healthy, rapidly growing plants over stressed ones. This hypothesis is particularly relevant for insects that specialize in young, actively growing tissues, like gall inducers, shoot borers, leaf miners, and certain aphids. Unlike stressed plants, vigorously growing plants are actively allocating resources to growth and the production of new nutrient-rich tissues. If an insect population can exploit these tissues, it can lead to higher survival rates, faster development, greater reproductive success, and ultimately significant population growth. One well-documented example of this hypothesis can be found with the arroyo willow sawfly, which is a gall-inducing insect that selectively oviposits on the longest, most vigorously growing shoots of its willow host. Females tend not to lay eggs on the slower growing or stunted shoots, as these offer fewer nutrients and consequently results in lower larval survival rates. The differentiation between the plant stress hypothesis and the Plant-Vigor hypothesis seems to depend on the type of feeding strategy that's employed. Sap-feeding insects and bark beetles are sometimes tuned in to exploit systematic physiological changes in stressed plants, while gall inducers or leaf miners can manipulate and take advantage of high-resource, fast-growing plant tissue. Now let's move up a trophic level and examine top-down regulation, which operates through predation, parasitism, and disease to shape insect populations. Perhaps at a super fundamental level, you have something like the enemies hypothesis that suggests herbivore populations can be regulated by natural enemies. One well-documented example of this is the spruce budworm, which is a major defoliator of North American conifer forests. Its population dynamics exhibit cyclical fluctuations, where periods of low population densities are maintained by strong parasitoid pressure from a variety of parasitoids. It's density-dependent regulation, more parasitism with higher levels of budworm presence, and it can act as something of a stabilizing force, keeping budworm numbers in check. But abiotic factors can actually complicate this. Mild winters can increase overwintering survival of budworm larvae, so budworm numbers rise faster than parasitoid populations can respond. This time-lagged response in parasitoid reproduction allows outbreaks to occur, overwhelming both natural enemies and host plant defenses. Predators are also key regulators of insect populations, often responding dynamically to prey abundance. In the case of the southern pine beetle, outbreaks are strongly influenced by its primary predator, the clerid beetle. The clerid beetle preys on adult and larva pine beetles, and under stable conditions, it can help to suppress pine beetle populations. However, like with the parasitoids, predator-prey cycles can be inherently time-lagged. The clerid beetle requires high pine beetle densities to fuel its own reproduction, meaning predator numbers rise only after pine beetle populations have already surged. This numerical lag often allows outbreaks to escalate before predators can adequately respond, creating boom and bust dynamics. These cases of top-down and bottom-up control can also be triggered by lateral interactions, meaning species within the same trophic level can modify the strength of predation, parasitism, or plant-mediated effects. Let me give you 2 examples. First, a case where bottom-up control is triggered by a lateral interaction. The winter moth and the green oak tortrix are both defoliators of oak trees. Winter moth larvae, however, hatch first and begin feeding early in the season, which triggers the tree's induced chemical defenses. This increases tannins and reduces foliage palatability. This now indirectly suppresses the green oak tortrix populations, which hatch later and must contend with low food quality. In this case, a lateral interaction shifts the bottom-up control, as the first herbivore altered the nutritional landscape, ultimately reducing the success of its competitor. Conversely, there can also be something called facilitation amongst herbivores, which can amplify population growth by enhancing host plant quality with a lateral interaction. So you can have the pine sawfly and the pine butterfly, which both feed on Scots pine. When the sawfly larvae feed on the pine needles, they trigger changes in nitrogen availability and terpene concentrations, which, rather than being a deterrent, actually improves the foliage quality for the pine butterfly larva. As a result, pine butterflies feeding on these same trees develop faster and survive at higher rates than those that feed on undamaged trees. In this case, a lateral interaction modified bottom-up regulation, making host plants more favorable rather than less favorable. These interactions complicate population dynamics. Sometimes lateral interactions can reinforce top-down or bottom-up suppression, and other times they can counteract it, enabling populations to increase. So there's often this complex and dynamic interplay of biotic and abiotic forces that shape insect populations. And these types of interactions don't just occur within a single system. They can extend across landscapes. There's something called the Moran effect. The Moran effect occurs when an environmental factor, such as precipitation, or temperature, or drought, acts across a large area, driving synchronous fluctuations in insect abundance. You could imagine that spatially independent insect populations over a broad region could be synchronized through broad-scale climate conditions. So climate can act as a broad-scale unifying force, synchronizing insect population dynamics even when local ecological conditions vary. These multi-trophic, multi-scale interactions highlight why population dynamics can be quite a challenge to predict. Simple models fall short because insect populations are shaped by an evolving web of interactions across trophic levels and environmental conditions. So why do insect populations change? There's no simple or single answer. Abiotic factors set the stage. Biotic factors then come in and act in a myriad of ways, and then you have interactions that provide a set of complex forces that shape populations in ecological communities. But understanding these mechanisms doesn't only help us know more about the natural world, it can also help inform management practices for things like agricultural pests or species conservation in the face of climate change. Well, that's all for today. Thanks for tuning in. I'll see you next time.